Hi, my name is Kyla, and I will be presenting on a locally found species and asking if moonglow anemones use fluorescent proteins to bait their prey. This is an outline for the presentation. I'll first be talking about the background information of moonglow anemones, their polymorphisms, how fluorescence works, GFP or green fluorescent proteins and their biological function, prey luring, and further questions as well as a conclusion. These are locally found moonglow anemones. You can see that they occur both in the green color morphology and a white color morphology, and the green color morphology is fluorescent under blue light. Even though these are found in the relatively same environmental conditions, one is fluorescent and one is not, which leads to the question if this is an adaptive color morphology or not. Anthopleura artemisia is an intertidal anemone of the family Actinidae. They're cnidarians and of the genus Anthopleura. Moonglow anemones range along the west coast of North America from Mexico to Alaska. And as I mentioned, they're found locally in El Katzimahow Sound, a UNESCO biosphere region. Moonglow anemones are found relatively high in the intertidal zone, and they're usually buried in sand or sediments, but they're also found on rocky outcrops. They reproduce both sexually and asexually through binary fission, and they're sessile benthic predators that entrap prey with nematocyst-loaded tentacles. Unlike related Anthopleura, moonglow anemones lack symbionts. And as I mentioned, they're found in two color morphologies, at least locally, the green fluorescent color morph and the white non-fluorescent color morph. In other areas, just of note, they can be found in other range of colors, but the green ones are the ones that are fluorescent, and that is due to the presence of green fluorescent proteins or GFPs and not symbionts. So under blue light, like you would find at high tide where the light is attenuated through the water column, or potentially even in the presence of bioluminescence, the anemones fluoresce. We know that this is due to GFP because they appear green. If they were fluorescing in more of a red range, that would maybe indicate that they do have symbionts. But in this case, we can confirm that the fluorescence is due to GFP. And you can see that the white color morph is non-fluorescent under the blue light conditions. So how does GFP fluorescence work? Well, fluorescence is an emission of light in a different wavelength of light than the fluorescent substance, in this case GFP, has absorbed. So GFP fluorescence is activated by the absorption of blue light and emits green light when the GFP is excited. Further to this, if you apply a yellow filter on the emitted green light, either for photography or in some organisms that may have yellow eye lenses or even pigments, that green emitted light appears much more vivid. The function of GFP is still actively debated. There are many hypotheses about the biological function of fluorescence. A very common one is the sunscreen hypothesis, which is that GFP is upregulated to protect the organism from damaging UV radiation. Another popular hypothesis is symbiont attractant, so that GFP is upregulated to attract symbionts such as dinoflagellates. And the symbiont protector hypothesis suggests that GFP is upregulated in order to protect symbionts that are photosynthesizing from harmful UV light. And a more recent hypothesis is that GFP could be upregulated to lure and increase predation rates. And this has been demonstrated in corals that are related to anemones. A recent study tested the abundance of GFP in 3 Anthoplura species that are closely related to moonglow anemones, based on the upregulation of GFP in 3 different light conditions. So they exposed the anemones to 3 different conditions: a dark condition, a tide pool condition with moderate light, and a bright light condition. And you can see that the abundance of GFP was greatest in the high light exposure conditions, lowest in the dark conditions, and moderate in the tide pools where the light was moderate, which indicates that GFP, at least in these 3 related species, is upregulated in the presence of light. However, interestingly, with the moonglow anemones, even when they're in the same lighting conditions, as is shown here, they can appear in different color morphologies, whereas the previous experiment would suggest that they might both be green and producing GFP, since this was a higher level intertidal position with quite a bit of UV light exposure, which you would find at low tide. At high tide, light is attenuated and is shifted to more of a blue range, and you can see where I've shined just a blue light on the anemones that the green fluorescence is suddenly much more apparent. And when you add a yellow filter, in this case to the camera, but if it was an organism with a yellow eye lens, or potentially even a dinoflagellate with light filtering in through their yellow pigments, you can see how much more vivid the fluorescence that's being excited by blue light is in the green color morph versus the white color morph, which is very low to no fluorescence at all. So a recent experiment with corals, where the researchers placed 3 treatments in a water column, a control with no fluorescence, green fluorescence treatment, and orange fluorescence treatment, that trapped plankton, resulted in green fluorescence attracting the highest levels of plankton. And within that sample, the majority of the plankton were copepods. While it's still unknown what moonglow anemones prey on, they do likely prey on copepods, which could be interesting in terms of the findings in coral that green fluorescence is preferentially attracting that prey source to them. In addition, other Anthoplura anemones are thought to prey and have been demonstrated to prey on planktonic copepods. Which leads to this further question: is the GFP that occurs in the green color morphologies of moonglow anemones upregulated in response to maybe low levels of food to attract prey items? So the incoming blue light from attenuation or potentially even bioluminescence could excite the GFP. The green light is emitted and potentially could preferentially draw in organisms with yellow lenses, maybe directly or indirectly, such as with dinoflagellates. Copepods prey on dinoflagellates, so this leads to the question if the anemones are using GFP as a lure to attract their prey, either directly or indirectly. Additionally, there are questions that come up whether these anemones are the same species or perhaps they're sibling species. Do the different phenotypes have different genotypes for producing GFP. Are they clones? Could they be resource partitioning to avoid competition? And what actually do they eat? And does that change based on the tide? While still very little is known about this local species, the role of GFP is also debated, but might be a good example to explore the role of GFP in moonglow anemones. Additionally, marine heat waves are increasing in frequency, and this could lead to changes in prey abundance. And that could lead to resulting changes to the upregulation of GFP in anemones. So GFP species like moonglow anemones could be biological indicators for disturbance. For example, they may be able to indicate a lack of prey through the upregulation of GFP. Thank you very much.